Hello, everyone, and welcome to Lytham Partners Spring 2022 Investor Conference. My name is Ben Shamsian, Vice President. Thank you for joining us for our cancer panel titled Technologies to Combat Cancer. We're joined by three executives from three great companies from the industry to get some insights on how they see this space. I've gathered some topics that are of interest to investors and hope to learn greatly from our panelists. Joining us today are Jeremy Svoboda, CEO of Perimeter Medical Imaging AI, Gerard Michel, CEO of Delcath Technologies, and Cameron Reynolds, CEO of VolitionRx. Before we begin with the discussion, I would like to have each of you introduce yourselves and provide a brief description on your companies. Jeremy, we can start with you.
Thanks, Ben. We're happy to be here. As you said, my name is Jeremy Svoboda. I'm the CEO of Perimeter Medical Imaging AI, and I've been a long-time medical devices, career-long medical device guy, and I joined Perimeter Medical Imaging in 2019 at a really exciting time for the organization. What Perimeter is doing is developing advanced imaging tools that provide ultra high resolution in real time advanced imaging to give surgeons the ability to solve a problem called intraoperative margin assessment. And what that means is when they're performing surgery on any solid cancer, what they try to do is remove the tumor and then a barrier of cancer-free tissue around the outside of that tumor. That's called a margin of healthy tissue. If they do not achieve that margin of healthy tissue around the outside, it generally increases the risk of recurrence. In the case of breast lumpectomies, it doubles the risk of local recurrence for patients. So instead of accepting that risk, the surgeon would call the patient back for another, sometimes a third operation. And unfortunately today, without Perimeter's technology, surgeons have to wait up to a week to get that information. What we're doing is bringing that imaging information into the operating room in real time, so the surgeon can have the information available to them, see if they need to take additional action on the patient right away, thus potentially avoiding all of the costs and complications with the additional operations. So really exciting time for Perimeter. We just went commercial with our flagship product late last year. And as the name implies, Perimeter Medical Imaging AI, we are in late stages of clinical validation on some advanced artificial intelligence to help surgeons interpret those images. So looking forward to sharing our story today, Ben. Thanks.
Thanks, Jeremy. Gerard?
Sure. Gerard Michel, CEO of Delcath. I've been here just 18 months within a couple of days, though it feels like it's been many years. It's been a very exciting time, a busy time for us. Delcath aims to be the leader in targeted, safe, and highly effective minimally invasive treatments for patients with cancers of the liver. And cancers of the liver, whether it's primary tumors such as hepatocellular carcinoma or metastatic cancer from another primary tumor, are a huge issue. Often the liver mets or the primary liver disease, but in the case of liver mets, become the life-limiting organ, the life-limiting issue for patients. And probably about 200,000 patients in the EU and the US every year have liver-dominant cancer. And it's very difficult to treat liver-dominant cancers for a variety of reasons. One of them has to do with the vasculature of the liver. It's very difficult to resect the cancer out of the liver. So more often than not, you can't resect or cut out the tumor. Secondly, systemic therapies have surprisingly low efficacy in the liver itself. And then even immuno-oncology agents, which are growing dramatically, they become less effective when you have the presence of a met in the liver, because the liver actually is an immune regulating organ. It actually makes your body more tolerant of cancer once you have a met in the liver. So it's a very large issue. Now, our first indication for our product is a very small ultra-orphan cancer called metastatic ocular melanoma. It affects probably about 1,500 patients a year in the US, about 3,000 in the US and Europe. The disease presents first in the eye. Once it metastasizes, though, it's almost always to the liver, and it presents in a very diffuse pattern where it's virtually impossible to ever surgically resect it. And it's a very chemo-resistant tumor. So it's an interesting proving ground in that it's one of the toughest liver tumor type cancers or mets you can go after. Our phase 3 data read out last year. We met all our primary and secondary endpoints. So we're very excited about that. We're putting our NDA into the FDA mid this year. The overall need for the product, though, goes well beyond ocular melanoma. There's a cancer called intrahepatic cholangiocarcinoma. That's about triple the size of that. That one almost always metastasizes to liver. But even cancers that don't metastasize a lot to liver, but they're really high volume, like breast cancer, it can become an issue. It's probably about 4,000, 5,000 patients a year with metastatic dominant breast cancer. Neuroendocrine tumors have a habit going to the liver. Pancreatic tumors almost always go to the liver. And the biggest one is colorectal cancer. Probably about 35,000 patients a year end up having liver-dominant colorectal cancer. And our product should theoretically work in all those cancer types. And there is some data out of Europe that does indeed work there. We've been approved under a CE mark in Europe for many years. That doesn't lead to a lot of reimbursement. I think a lot of the listeners may know that the CE mark is a relatively low bar for approval. A lot of devices get approved under that. We are actually a combination drug device. The device limits the exposure of chemo to the liver, and it filters the chemo out of the blood prior to it going systemically. So we got approved in Europe as a device. We're being regulated as a combination drug device in the US. So we're excited about getting the NDA and getting approved in ocular melanoma, but also expanding development along many other tumor types. And hopefully at some point we might become the mainstay for treatment of mets in the liver.
Thank you. Cameron?
And welcome everyone, and thanks Ben and Jeremy, Gerard, very nice to hear all of that. Volition, we're listed on the New York Stock Exchange, VNRX. We're a cancer diagnostic company, but what we measure are chromatin fragments. First of all, my background, I was one of the founders of the company, I guess 12 years now, time flies. We've spent these 12 years, I think very wisely, really developing a very strong, stable platform to measure the epigenetic signals in circulation, most notably in blood. Why do they matter? We were set up on the very simple idea that epigenetics, which means above genetics, are going to be incredibly important in diagnostics. Think of it this way, the genetics is your DNA, huge amount of money, probably many hundreds of billions spent trying to measure the DNA signals, the genetic signals. The epigenetics, I think, are, if not more important, and have really been kind of overlooked up until quite recently. Our blood tests, so measure the nucleosomes or chromatin fragments in circulation. Our tests are very low cost, very easy to administer. They're ELISA-based, so therefore they can be run in any lab in the world. And we're developing the platform. It took us about 10 years to get it very, very stable, very reproducible, very reliable. What really makes us different is our blood tests are truly routine. What does routine mean? It means it can be run in any lab in the world at low cost, and as importantly, a small amount of blood. So we've developed a fantastic intellectual property portfolio, which really dominates what we do, which is nucleosomes and chromatin fragments in circulation. And now we're commercializing in a wide range of areas. These signals are quite different in different cancers, and also in a wide range of other diseases, including things like mitosis, which is what causes death in COVID and sepsis. And actually, they're preserved through species. So we have a subsidiary now, which is just launching its first products worldwide in animals, detecting cancer in dogs to start with. So we have a fantastic platform which can measure very simply, very quickly, very cheaply these changes in circulation. There are hundreds of different changes of the epigenetics on the nucleosomes, and theoretically we can measure a large number of them. We've got about a dozen we can do now, and we're putting that together to measure a range of different cancers, including colorectal, the blood cancers, lung cancer, and as I said, the blood cancers in dogs. We've already launched our first products. So it's a very exciting time for us. We're in the process of commercializing this incredibly useful and very versatile platform. And in the cancer space, I think we can be a very, very disruptive and differentiated, because we are the only truly low-cost, very democratic platform that can be used in a very wide range of cancers. So expect to see a range of products from us.
All right, thank you, gentlemen, for that insight. each of you play in a different area of combating cancer. What are the alternatives to what you bring to the table, and why is your technology superior? Gerard, I'll begin with you.
Sure. So right now there are basically three ways to treat patients when they have tumors in the liver or liver mets. One is systemic chemo, which is commonly done for things such as colorectal. Unfortunately, the efficacy of actually, getting a decent response in the liver is pretty low for systemic chemo, and you subject the patient to the systemic effects of chemotherapy, which can be pretty damaging and really lower the quality of life. If you're not going to do systemic therapy, the two regional or local therapies that are done are Transarterial Embolization, or TACE, and Y90. In the former, with Transarterial Embolization, either a viscous substance or beads are used to combine with chemotherapy. And then you take a catheter, and by embolizing the tumor with that substance and the chemo eluting out, it generally kills or shrinks the tumor. And there are about 50 to 60,000 of those treatments done per year in the US. A second option is something called Y90, yttrium-90, and those are radioactive beads that are delivered to the tumor, again via a catheter. And there are about 10 to 15,000 treatments of those per year in the US and growing. Both of those treatments are reasonably effective for the single tumors they get at, but tumors generally recur and then retreatment is limited. For TACE, you actually destroy the vasculature leading to the tumor, so when they regrow, you can't retreat. With Y90, it's very toxic, so you're limited to 2 treatments. So if you have multiple tumors and you only treat one lobe at a time, you can't treat multiple tumors with Y90. If you can't see the tumor, you can't treat it. And often if you have one met, you have multiple metastases, even if you can't see them. Again, these treat tumor by tumor or lobe by lobe. So what you can't see, you can't treat. So it's important to be able to treat those micrometastases, and you can't. With our product, what we do is we actually treat the whole liver with high doses of chemo, levels of chemo that would be potentially deadly if you didn't have our system. Hepatocytes are very, very robust. They're the garbage disposal cells of the body. So you can put very high doses, in our case, we use melphalan, into the liver. And what we do is we block the blood coming out of the liver with a double balloon catheter. So the blood that comes out of the hepatic vein is trapped, moves out of the body via catheter, and we filter it via a proprietary filter that absorbs well over 85%, usually in the high 90% of the chemo out of the blood before the blood goes back into the patient. So we're able to isolate the liver, treat with very high doses, and minimize systemic side effects. So we can, treat the whole liver and treat repeatably, and it's minimally invasive. The competitors out there, TACE and Y90, they can't do that. They work reasonably well for hepatocellular carcinoma, but it's a primary liver cancer where the tumor shows up as a single tumor. But for the over 100,000 patients a year who have mets from other tumor types that generally show up as many, many small mets, often these treatments are not terribly effective. So that's the difference between us and Y90 and TACE. We can treat repeatedly, we can treat the whole liver, and we don't actually have to see the tumor. And it's interesting hearing a little bit about Volition. We're very interested actually in various types of circulating tumor DNA or circulating free DNA assays, looking for those that might be able to signal a liver met before you can even see them. And I know there's different methylation patterns actually for liver mets than the primary tumor. So maybe we'll chat a little later on, Cameron.
Yeah, they absolutely are. I'd love to.
Cameron, please go ahead.
Oh, yeah, just on that actually. So we have several aspects to our platform, our own test, and also we call Nu.Q Discover, where we can use assays for methylation patterns, as you say. Also, netosis is a very big driver in rejection and a whole lot of other issues. Assays measure netosis almost perfectly. And so they can be used as a primary diagnosis, also for monitoring, and also a lot of other things. So certainly worth a follow-up. So we're just in the cancer space. What differentiates us on our cancer platform? Most cancers are diagnosed through some level of imaging or looking at it, if you will. Mammography is looking at a scan, colonoscopy, endoscopy, skin cancer, you have a look at it. And those obviously suffer from a lot of, they can be invasive, they're interpretive, they can't be used as easily to mass screen like a blood test. So I think everyone agrees if a blood test would work, a blood test would be the best. The problem has been, there's been none that really have worked very well. Certainly not for screening. The PSA is kind of all right, but everyone complains about it, but it does its job to some level. There's a sort of more background tests like CEA. There's been a real push quite recently for more accurate blood tests, so quote unquote liquid biopsies. The issue with those, although some of them I think may end up working quite well, they're the opposite end to what we do. They're very expensive. They're they're not routine. They need a lot of blood. You could be talking thousands of dollars. So that works for some people. That doesn't work for others. And it's not going to be a routine screening test around the world for all those reasons. So we're the only ones that truly have a routine test that can be the frontline test for further diagnosis from screening. And it's an incredibly versatile platform that's we're far cheaper, far easier, far quicker, a tiny drop of blood. So Ben, if you were in the doctor's office, they could just tick an extra box with all the other tests that you do, goes to the lab like any other test. And our cost of goods are quite low. We're producing the key components, the nucleosomes and the controls. And we aim to be in the blood tests in the US somewhere in the $100 range or lower, which still gives an extremely good margin. In the pet world, that's about half that price, about $50, so that it can be very affordable for the average pet owner as well, for the cancers we can detect very well. And as Gerard said, our platform, apart from netosis, which I think could be even bigger than the cancers, it's a driver. We don't actually measure the cancer, we measure chromatin fragments, which are also encompassing the DNA. So we can also enable all the liquid biopsy companies by concentrating. We can enable companies such as some ones here, where we can help with methylation patterns. Ours detect that extremely well. And we have a whole new program going of Nu-Q capture, where actually it's quite complicated patterns on the methylation patterns we can measure, either through sequencing or through mass spectrometry, where we just got early this year, late last year, we got a paper published in Nature Science, one of the smaller Nature journals, incredibly well thought through paper. So the epigenetics is really on the cusp of breakthrough. And if you look at liquid biopsy companies, which really all used to talk about DNA mutations, it's all become about methylation patterns now, which is of course epigenetic. And they still use sequencing to measure it because that's what they do. I mean, if you're owned by Illumina or you sequence, that's a very smart thing to do. But I do not think the ultimate answer for everything is sequencing and genetics. I think simplicity and epigenetics are a big part of what we do. So I think Volition's got a very strong future, particularly now we've done the hard yards, got the platform really stable and really strong. It can be used in many, many areas. So I think we could not be more different from anyone else in the diagnostic space, particularly in the blood space, given all the things I said.
Thank you. Jeremy?
Yeah, thanks, Ben. Yeah, and it's interesting to hear these two different stories. I frequently say, if you think about the recent history of trying to combat cancer, there's been a lot of capital allocated to early detection and better detection, and then there's been a lot of capital allocated to the medical oncology side. Meanwhile, where Perimeter fits in this story is in the surgical oncology space, where there's still a large opportunity for innovation, especially where our initial target market is the breast lumpectomy market. And, there's from a substitute perspective or an alternative to Perimeter, one of the things that surgeons resort to is just cutting out insurance tissue. So they know that this is such a big problem. It happens, anywhere from 20 to 25% of the time in breast lumpectomy patients. So one of the, surgical guidelines is to go back into the patient after you've cut out the primary specimen and you think you've removed all the tumor, go back in and take some insurance tissue from each part of the cavity left behind just to make sure you got it all, which is, far from what we want to provide to our patients, particularly in a space where cosmesis and post-op sensation are so important to patient quality of survivorship. And at the other end of the spectrum, what we have is, surgeons are typically x-raying these specimens to begin with anyways. So, pre-operative in the planning session, they'll put some navigation material in, usually a guide wire or a radioactive seed to help them navigate in the cavity while they're performing the operation. And then to ensure they've removed all of those foreign objects from the patient, they'll x-ray the specimen and ensure anything that went in came out. And in the case of breast lumpectomies, one of your body's reactions is to create little microcalcifications, tiny little calcium deposits, the same things that show up in the imaging that Cameron was just talking about in a mammography. You'll see a cluster of calcifications, and the surgeon will resort to, when they're x-raying, to ensure they've removed all of the navigation material. If they see any calcifications at any of the edges, at the perimeter of the specimen at all, they'll use that as a proxy to go back in and target those shaves that I just talked about. The problem with this is you're not seeing the disease itself, you're seeing a symptom of the disease. So you end up with a really low level of specificity, end up taking unnecessary tissue again as well. And, in the case of perimeter, we have, resolution 10 times that of X-ray to actually give the surgeon the ability to see these microscopic diseases such as ductal carcinoma in situ, which is, similar to what Gerard would say is, it's, you can't treat what you can't see. And in the case of X-ray, you can't see it. And then on top of that, it tends to kind of bounce around. It's not contiguous as like invasive ductal carcinoma would be. So, we feel like we give the surgeon, kind of a unique perspective and unique information to help them make informed decisions as they're treating their patients in the OR.
Right, thank you. All of you have gone either through FDA approval or in the process of it. Tell us about the experience there. What have you seen from the FDA in recent months or years? Has it become easier or more difficult to sort of navigate the process? Jeremy, I'll begin with you.
Yeah, I think we've seen, and I'll talk about the FDA specifically, but, in Gerard's case, he mentioned the CE marking, and that, I think, historically we've kind of said, the CE mark model is, a little better because let's get this new technology out to the marketplace, do a more robust method of post-market surveillance, and get patients access to this technology, but also make sure it's safe and effective. I think ironically they're kind of moving in the other direction with some of the recent changes in regulation over there. But for us on the FDA side, I've seen a little bit of the opposite, where I think particularly I'll touch on our AI indication, because this is something that the FDA is really evolving and really partnering with industry to try to figure out the best path to market. And for us, we haven't been cleared with the AI version of the technology, but they did grant us a breakthrough device designation, which was a really big tailwind for us from a regulatory perspective. And it gives us access to, have dialogue with the FDA about, okay, what are some ways we can get this to market quickly, safely and effectively, but quickly. And in the case of artificial intelligence, we know the more data we accumulate in the commercial space, treating patients, the smarter and better our algorithm will get. So how can we really work together now to get this technology to market quickly, but have a predefined change control process so that as the model improves and grows, we can continue to incorporate those updates in the least burdensome path so that patients can have access to that technology going forward. So, I'd say, particularly with this new evolving space, in what computer assisted diagnostics or, that particular area, I think they've been very willing and eager to partner with industry to find ways to get their technology out to patients.
Thank you. Gerard?
It's hard for me to say whether or not we've seen a change in the FDA becoming easier or more difficult. I think as individual CEOs and management teams, you live in your product, your set of interactions. So I do hear investors complain about that, but at times it's really just, if it happened to be in a company where there was an issue with the FDA. We've had an interesting history with the FDA. Delcath has been around a long time. This idea for our product came up in the 90s, I think. And it was based on a surgical procedure where people were opening up the patient's gut and basically clamping off the liver, putting on a heart-lung machine, a perfusion circuit, dosing with very high doses of chemo, flushing the liver with saline, and then knitting everything back together. And the procedure itself had great response rates across numerous tumor types, but about 5% or plus of the patients would actually die from the procedure. So that never really took off. And a bunch of docs had an idea about doing this minimally invasively, but it never was, venture back. It didn't have a very solid management team behind it. They got pushed to the clinic too quickly. They never had a real collaborative approach with the FDA, and without going through all the details, it resulted into a CRL almost 10 years ago. And it took the company that long to get back on its feet, rerun the trials with a device that was fit for purpose, well designed, with a design history file, everything manufactured, GMP, et cetera. And we have a great relationship with the FDA now, and I think that's because the company was so, chastened from what happened years ago. We really bend over backwards to try to listen to the perspectives and have an open dialogue. You can only sell so much with the FDA. Your data is your data. Thankfully, our data is great. But, in terms of whether the FDA is getting more difficult, I don't know. I think it just depends on your particular experience at the moment. Obviously, they're, an incredibly important factor in the company's success, and it's best to be as open with them as possible, be as collaborative with them as possible. And spinning the data a little bit maybe, but don't fool yourself. Look up the data every single way to Sunday. The best thing to do is to be forthright and appear that way.
All right. Cameron, any thoughts there?
Yeah, I think a little bit what both speakers have said before. So we overall, we're an international company, so we have a lot of options where we get regulatory approval. Obviously, the big ones are the ones you mentioned, the CE mark in Europe and the FDA. Our strategy has always been to do the other countries first and move to the FDA, which we've done and we're starting now. And I think the CE mark, I think Jeremy said it's right, they're heading the other way from IVDD to IVDR, where it's more regulatory work. So I think the CE mark used to mean you could sell it, but didn't mean someone would buy it. Where the FDA approval kind of means you are at best in class or at best at something. So I think we're very, very serious now. The platform's stable in launching a range of products, animal, Europe, and US, and we are multinational. And I think you can kind of do different things in different areas, and different countries and different regions need different things. The US is a lot less cost sensitive, but very sensitive to accuracy, where it's a real cost benefit in most other places in the world. So we are getting very serious about the FDA. We do have some trials underway and in process, and I think the advice Gerard said was a very good one. Hire the best people who know what they're doing, and don't push something that's not going to happen, because we just raise expectations and then it collapses. I don't think they can be fooled, if you know what I mean. So and you're usually fooling yourself as much as anyone else to start with. So I think the FDA, I think the emergency use application actually is very helpful. We obviously were involved in a lot of work on NETosis, which is the driver in COVID. So it could be a strong option for us going forward. But I think particularly small companies, the temptation is to try to kind of wedge a small trial into something bigger than it looks. But ultimately, I think there's not too much to do except a big proper trial with very good consultants. And it's expensive, but it'll save you money in the long run. Trying to do it on the cheap with something which is suboptimal normally does not succeed, and you end up wasting a lot of time and money. So, and that's what we're in the process now of doing. I think feedback from groups, it is more difficult now if you're not in emergency use. There is a backlog with the FDA because they have been so COVID focused, which is, I guess, understandable given everything. But so our feedback would be, if you can find something that can get emergency use, then it's very much worth doing that. But if you're going to do it the other way, do it properly and get all the work you need to do, and do it quickly, because no one likes time, but make sure it's done properly. And I think then the FDA will be what you need it to be.
Great, thanks. let's talk about the insurance companies. Help us understand the process you have to go through to make sure your product is covered or partially covered by insurance. Gerard, I'll begin with you.
Sure. So our product's first indication, metastatic ocular melanoma, is an ultra-orphan indication. One of the upsides for that is that you never really hit the radar screen for payers, whether it's CMS or private payers. This is a disease that, right now our data, the survival data is not yet fully mature, but it looks like we're doing about a 6-month advantage for these patients in terms of longevity. That's pretty meaningful. And given the rarity of the product, I think we won't have too much trouble getting reimbursed. Now, with that said, we don't want the oncologists or the office to have to do one-off calls every time to get pre-approval or have to go back and forth. We'd like to make it as simple as possible. So there are some things we have to do and will do to try to smooth the process. In the US, I think you can kind of view reimbursement for hospital type products, whether it's drug or device, kind of in a 2x2 matrix. Is it private pay or is it CMS? And we happen to straddle that in terms of our age groups for most of our patients. And the second question is it an inpatient or outpatient? The majority of our procedures will be outpatient, but there are some set that will be inpatient. Outpatient for CMS is kind of a midnight to midnight rule. So if you don't spend 2 nights in the hospital, you're okay. We expect most of our patients will spend a night. Some might spend 2. So we have to look through, all 4 boxes there, which makes life a little more complicated. But again, whilst we're working through all the reimbursement processes, we do know that patients will get approved to get treated because this is a deadly disease. But for the private payers, this will get paid under a J code if they're outpatient. So it's basically get paid as a drug. We'll use a temporary C code. Then eventually we'll get a J code. They'll have carve-outs in their contracts where these oncology companies, hospitals do it. Most of our treating centers are going to be dedicated oncology centers that are quite efficient, we'll say, or proficient in terms of getting reimbursed. They know how to work it. So that helps for us. We're not going to have to deal with community oncology centers. But eventually what we'll have to do is apply for a new CPT code. We have that in the works. We're going to have to apply for a HCPCS code again. That's a separate J code that'll be a pass through. On the inpatient basis, we were going to have to apply for an ICD-10 PCS code, and then eventually, once we have that, a DRG code. Leading into that, to help the payers, we're going to apply for a new technology add-on payment, an NTAP. That gets added to the DRG bundle payment. But the ICD-10 codes and DRG codes, we're prepared to go after those. And the last thing is you kind of put together something called a value dossier to show primarily to the payers. And in that case, what you're showing is either, hey, we're on an efficacy basis on, let's say, dollars per month of extended life, whatever metric you want to use. You're ideally in the same ballpark as the other therapies. Or if you're not, it's best to be ultra orphan like we are. We will be well within the grounds or better than the high-end oncology agents used to treat our disease. If they put us against regional therapies like PACE or Y90, which don't have much efficacy data, we're going to look very expensive. But because it's so rare, ultra rare, again, it's not going to hit the radar screen. And number 2, there are systemic therapies that are far more money than we are or likely to be. And we'll have a label. The other regional therapies don't have a label to treat this disease. So I think we'll be in pretty good shape.
All right. Cameron?
So we're still in the process of the trial. So I can just give you a little background. Obviously, we started in the vet space for the first cancer products. That market is not insurance-based pretty much at all. So it's mostly user pays by the owner, not the dog, of course. So ultimately, it's an out-of-pocket expense, which is much easier to do. So, but because our tests are in that $100 or sub-$100 range, it's something which could easily be, you can do with a CPS code, or you can put together the panel of different assays. We're in the process of working that out now. But our whole strategy is to keep it a very valued product so that it doesn't become painful for anybody. I think we could be something where we're doing tens of millions of tests per year for each test. So, you know, I think to get there, you need to make sure that it is very, very good value and something can be used without someone really having to put a big decision into it, particularly in the monitoring when it has to be perhaps run 10 or 20 times during the treatment. So we're in the process now, but I think given the fact we take money out of the system, we save a lot of money, obviously, in late-stage cancers or the other diagnosis. Given we're very low cost and routine, no special equipment, we'll go through a slightly different process of insurance reimbursement, but that's something which is ahead of us, so I cannot go through too many of the details now.
Thank you. And Jeremy?
Yeah, so I'd say similar to what Cameron was just saying. So we're kind of, breast lumpectomy specifically are part of what's called a comprehensive APC within the reimbursement landscape. So from a CMS perspective, you know, it's already kind of a bundled payment, if you will, for everything that goes along with it. And, you know, the problem we're helping to solve is a very expensive problem today. So you heard me mention earlier, it's impacting, you know, about 1 in 4 patients, you know, that require a completely separate operation to treat this problem. So you've got about 1 in 4 patients coming back for an additional $16,000 in treatment costs. So we're talking about a $4,000 cost per patient here that we're, you know, helping the payer avoid. And this is really squarely in the direction that, you know, we're moving from a value-based care environment. So instead of a fee for service, we want to have a pay per episode of care right along with that bundled payment. So some of the programs you've seen in the cardio world or in the orthopedic world where, you know, if you have to come back and revise, you know, it's still part of that first payment. That's squarely, you know, the kind of target market we'd be going after in these early days. You know, think the integrated payer providers or the value-based care organizations. Meanwhile, while we're commercializing under that model, we do have a temporary CPT code to be used today, help us track utility, help us track the, you know, the savings to payers all along that way. And, you know, once the data is there and after our clinical validation study on the AI, you know, we would expect that we'd be able to upgrade that to a category 1 code and get a specific facility reimbursement for that technology as well.
All right, thank you. We're coming up on time, so last question here. Can each one of you tell me something that either your customer base or your investor base may not fully appreciate about either your company or your technology? Cameron, I'll begin with you.
Just the tremendous versatility of the nucleosomics platform. I mean, the chromatin fragments are incredibly important in cancer. They're incredibly important in cancer detection. They're incredibly important in NETosis, which is the biggest killer of people if you add up sepsis, COVID, influenza. NETosis is a big driver in metastasizing cancer, and the epigenetic signals are incredibly important. So we've developed a platform now, and it can measure the fragments on a microtiter plate, on a lateral flow, on a small machine, point of care, large HA machines we're working on now as well. So incredibly versatile, and it's a platform of platforms. So I think if you'd asked, I guess, anyone about epigenetics a few years ago, it was still the poor cousin of genetics, and it was sort of how cute, you know, DNA is the answer. Now, I don't think anyone thinks that way at all, and particularly after NETosis has really come through and shown how important the nets are in pretty much everything. So I think there's a growing realization how important all this is. And I think we have the absolutely ideal platform for all our own things and also to license out and to let others use. And I think given all of that and given it can be used pretty much anywhere in the world, in any lab, on any person, given its low cost and democratic nature, I think flexibility and versatility and worldwide reach are something which is only just now being understood. So hopefully it'll become more understood and we can really launch some great products.
Gerard?
Sure, I think similar to what Cameron was saying is, you know, the utility, how broadly this technology we have could eventually be used. Most people who die of cancer don't die from the primary tumor. I think brain cancer aside, it's usually the mets that kill them. And the lung and liver are probably the two, and bone are the three places that you usually get mets. Some tumors have a habit of just going or predominantly going to the liver. And that often becomes a life-limiting organ for patients. And we're talking, a good 100,000 plus, at least in the US every year, that probably fall into that category. The system we're using, we use an alkylating agent that's pretty much any rapidly growing cell it kills. That's part of the reason you need to use our system, in that it would wipe out your bone marrow if you use the doses we're using of melphalan. This product is derived from the concept of intrahepatic perfusion. That was that surgical technique I described a little earlier on. And there's a ton of data out there in CRC and other tumor types that shows really good response rates. Now, the FDA, being the FDA, they're going to want to see data in every tumor type before we would get a label. We'll certainly expand beyond ocular melanoma for our label, but we aren't necessarily going to go after every tumor type. We just need to generate enough data to get on guidelines, and that'll be sufficient, I think, for the business. So we're looking forward to starting those new trials and expanding well beyond just ocular melanoma. So I think that's something investors miss. They see this very small initial indication that maybe could generate a few $100 million in revenue in the US and Europe, but it goes well beyond that.
All right, thank you. And finally, Jeremy.
Yeah, well, third time lucky. I'll stick with the same story, because I think your question made me chuckle a little bit, because our customers fully appreciate our versatility. It's just not quite yet translating to the investor base with us. And as we introduce the technology to a new general surgeon or a pathologist sees it, I mean, instantly the wheels start spinning, and they're very excited about what other indications they could be using our technology for. So I'd kind of pile on to what both Cameron and Gerard said, and I think we're really excited about 2022 being a bit of a transition year where we start showing some meaningful progress towards I've kind of dubbed it the horizontal expansion into other tissue types, but also, across the surgical continuum of care, we think we can really assist with, effectively an optical biopsy that can give the radiologist an instant, feedback on whether or not they penetrated the targeted lesion or what that lesion looks like. And also post-op in pathology, unfortunately, it's a gold standard today, but it's also a sampling technique. So when they're looking at these margins, they tend to only look at about 16% of the margin. So, technology like us that can give them a gross pathological assessment in real time to help them target the right 16%, I think adds a ton of value to that part of the workflow as well. So looking forward to, getting some clinical data going and, harnessing that feedback that we're getting from our customer base and turning them into clinical investigators to help us develop in those directions.
All right. Unfortunately, that's all the time we have. I want to thank all of our panelists, as well as for everyone for listening. If any investors would like to get in touch with any of our panel companies, please reach out to myself or any other members of the Lytham Partners team. Thank you, and have a good day, everyone.
Take care. Thanks for your time.